What is the chance a 40-year-old today could live forever?
So I think that the chance for a 40-year-old today in the Western world is more than 50%.
What if longevity technology doesn't just save Gen Z, but also Gen X and even the boomers, making 150 years or more a real possibility? That's what we explore today on Mind Blowers with Aubrey de Grey. What is the likelihood of a 40-year-old person today who's in pretty good health that person could live forever, or at least not die from aging?
Right. So not dying from aging is, of course, the point, because I'm not working on stopping people from being hit by trucks or stopping the planet from being hit by an asteroid. Though, of course, I'm very pleased that other people are working on those things. So I think that the chance for a 40-year-old today in the Western world is more than 50%. I have said that I've been giving time frame estimates for how long it's going to take to get to LEV for some time. And my current estimate is 12 to 15 years, in other words, late 2030s. And the people who will benefit from this kind of technology when it comes along will be people who are typically in their 60s at that time. So that means someone who's 40 now has a good chance.
And how about people in their 50s or 60s? Do they stand a chance?
Yeah, I mean, so first of all, let's be clear that the major uncertainty here is how soon we will actually reach LEV. That's a point in time that one of my colleagues has called the methuselarity, by contrast with the singularity. The idea is that, I think it's 50% likely that we'll get there in the next 12 to 15 years, but there's at least a 5% chance that we won't get there for 100 years if we just hit a whole bunch of, unforeseen obstacles. So we have no idea, really. However, what we can certainly say is that one's chances of benefiting are not really determined by one's chronological age at all. They're determined by one's biological age. Essentially, even the kind of first generation rejuvenation therapies, which will probably be in various ways quite clunky, they will work for people who have got maybe 10 or 20 years life left in them. But they'll probably be too strenuous for people who are already at death's door. So, if you're an average 50-year-old, you've got a pretty good chance. If you're a better than average 61-year-old like me, you've got a pretty good chance. But it's only a chance. We can never know with any pioneering technology.
But what would kind of be the top 3 pieces of advice you would be giving to people who would like to, be there for LEV and to keep their biological age low?
Well, the 3 types of advice of the kind that you're looking for are very boring. They are essentially, they come down to live the way your mother told you to. Don't get seriously overweight, don't smoke, get enough sleep, have a reasonably varied diet. But of course, the real answer is that because the issue is how soon it's going to happen, one of the biggest things you can do is to try to make it happen sooner. By, obviously, some people are wealthy enough to be able to make a difference by supporting the research financially, and I spend a painful amount of my time raising money raising small amounts of money to do the most important work. Some people will do what you're doing right now. They will get the word out to the wider world by interviewing me or whatever. People who are going into biology, for example, as youngsters, can pick what field they go into on the basis of its impact on all of this. Pretty much anyone can do advocacy of one sort or another. They can, talk to their friends, their family, their colleagues. When it comes to financial contributions, I always say, first of all, every dollar counts. We're going to always have more small donors than large donors. But also, I want to emphasize that, that the poorer you are, the more people who are wealthier than you. So that, again, brings it back to advocacy. And honestly, if one does the arithmetic, one can make more of a difference that way than one can by, living the way your mother told you to. Quite apart from the fact that one will make more of a difference to everybody else, not just oneself.
Is there a middle ground where you haven't reached longevity escape velocity, but you kind of have fixed many of the time bombs in the body, so you have, you could say, a reduced rate of aging? but not kind of stopped it?
Probably not, actually. In short-lived species like mice, we will never reach longevity escape velocity because the idea is to fix things faster than time is passing, to get better and better at fixing things. And you just don't have time in mice. But in really long-lived species like humans, it's the other way around. Essentially, once we get about 20 years of additional life, that will be enough. That will be the methuselarity. We will not have any trouble keeping up after that. And it's hard for me to see how we will get, let's say, 10 years of extra life without doing pretty much all of what we have to do to get 20. In other words, we have to get to a certain level of comprehensiveness of the treatments to be able to knock back all the major causes of death. If we remember that, for example, eliminating cancer completely would only give us like 2 years of extra life, 2.5 maybe. And eliminating heart disease would be like 3 or 3.5. So going up to 10 is really hard. And I think when we get to 10, we'll be at 20 and pretty much at the same time.
And so what if you look at the individuals, for example, who are participating in, I think it's called the Rejuvenation Olympics, like Bryan Johnson, for example. So, for example, I think he says he's quite confident that he will live to his 150. So it's not clear to me whether he believes that would be including tech that will be available when we have longevity escape velocity, or whether that would be alone with kind of the data and his habits. Are you aware of that?
Yeah, honestly, I'm getting a bit frustrated with Bryan. I know him quite well, and he says wonderful things about what we're doing. And I certainly believe that he is, making a big contribution to raising awareness and enthusiasm for doing things about aging. But he isn't being at all logical about this whole thing. He's not, for example, putting any of his own money into research. He's putting it all into, his own treatments and disseminating these, improvements in how to use what we can do already. And he must understand, because everyone's told him that this will not give him, this will not get him to 150. It won't get him to 100. The fact is, he must know that. So I kind of, I'm looking forward to getting him to come clean, so to speak, with regard to the question you're asking.
Can you explain to me? I know there are different ways to measure biological aging, right? And when you look at these rejuvenation Olympics, to me it seems like you have some people who, according to that website, only age with a speed of 58% or something like that. And it also seems that some people have become younger biologically by changing habits. So let's say you had a biological age like at 50, and then you do everything right, and suddenly it drops to 45. Do these results make sense, or are we more kind of tricking ourselves to believe we're becoming younger?
A good question. It's a very important question. Of course, the wonderful thing for our work at LED Foundation is because we're working in mice, we don't have to worry about any of this. We can just measure longevity and we're done. Of course, we are measuring all manner of different aspects of health as well. But honestly, the acid test of longevity is always going to be the thing that matters most in terms of convincing people that we've made progress. And we can do that. Whereas, of course, with humans, we can't, because it takes too long. So, so the measures of biological age that are so fashionable right now, these epigenetic clocks and so on, still have a long way to go to be really validated, I believe, at least. At the moment, what we have is clocks that can not only tell you how old you are chronologically, which you already knew, they can also, to some extent, tell you how soon you're going to get sick. So in other words, there's some degree of measurement of biological age. However, what we need, and really do not have yet, is clocks that robustly reflect the impact of interventions. And here I don't just mean high-tech interventions, stem cells or plasma exchange or whatever. I mean lifestyle interventions, anything. At the moment, there's just not been enough data. We've got plenty of data on a variety of different people, lots and lots of people, but it's just going to take a long time to gather data on people who have been availing themselves of different interventions and seeing what happens to their biological age as measured by these things like epigenetic clocks or whatever. At the moment, therefore, I believe that the gold standard for biological age is the same as it ever was. In other words, we should measure functional things, physiological things like grip strength and treadmill performance and so on, and cognitive things, of course, as well. Memory, such like, which definitely people have got pretty good at identifying ways to measure these things that are reflective of age, of biological age, because they do actually change at a respectable rate well before people exhibit anything that would be called a disease of aging. I don't have encyclopedic knowledge of how the biological age is being measured in these rejuvenation Olympics, but I would say that things like that are what should be getting measured. Now, to come back to your question about bona fide rejuvenation as a result of living right or whatever, I think I definitely do not want to downplay that. I think it's very important that people should do their best to keep their biological age as low as possible and have it increase as slowly as possible. And so people may think, well, hang on, I thought that damage repair was the only way you could reduce your biological age, and we can't do that yet, to speak of. So this is a little subtle. We have, of course, built into us a huge arsenal of automatic built-in damage repair machinery that eliminate damage as metabolism generates it. And the kind of damage that we automatically repair, I do not even include in my definition of damage. I only include things that we don't have machinery to repair and that we need to develop medical machinery to repair instead. But it's there and it accumulates. And it accumulates in a manner that is not necessarily optimized, because the body doesn't really care about living beyond the point where it's done reproduction. So essentially, if you do things that might not necessarily be called damage repair, but they are good for you, like, you lose weight or whatever, then you are somehow, you are taking the pressure off these various damage repair mechanisms that we already have built into us. And that means they will work better. So you will see a rather widespread, rejuvenation by some measures as a result of things like just getting into a better state of fitness. But whether we should call that bona fide rejuvenation in the sense of reducing your biological age, it's questionable, because you really shouldn't have been overweight in the first place, is really what I'm saying.
But I mean, if I understood Bryan Johnson correctly, for example, what he said is, there's never been a human in history like me before, because I measure everything and I adjust to whatever is perfect. So if there was a lady who could live till she's 120, I should be getting 20, 30 extra years because of that optimization. And that is kind of what you're saying. That is not happening because the body is not built for that.
Well, it's more than that. The first thing is that Bryan is not built like Jean Calment. First of all, he has a Y chromosome. Men tend to live less long than women in the first place. Secondly, there are all manner of other genetic differences between Bryan and Jean Calment that, are by and large going to be life-shortening, because Jean Calment was selected, was the one person who had the right genes, as well as presumably getting a lot of other things right. Against that, though, the fact is, if the world were populated entirely by people who had exactly the same genetics and the same life history as Bryan Johnson, then he'd be absolutely right that, the fact that he's doing such an enormous amount of measurement and adaptation of his regime in response to those measurements is an extremely good thing. And, if there's one thing that I would take away, I would say we should take away from what Bryan is doing as a lesson to all of us is that measurement is really, really important. And actually, in some ways, it's easy not to do that. It's easy to look at what Bryan is doing, his regime and everything, and say, oh, I should just have that regime. That's nonsense. That's not what Bryan is really telling us at all. He's telling us, find out what works for you. And different things work for different people. So he talks about sleep an awful lot. Now, there's no question sleep is really important. You've got to have enough sleep. But what he talks about is more than that. He talks about sleep schedule. He places a huge emphasis on getting the same amount of sleep at the same times a day, every day. In fact, there was a long period of time when he simply refused to do any travel that involved crossing more than 2 time zones because of the disruption. And he's relaxed that now. It's just like there's too much to do. But he used to do that. And he found that's because he, through all the measurements he was doing, he found that was important for him. But honestly, that sleep schedule rigidity is much less important for a lot of other people. So I, for example, I definitely need my sleep, but if I have it disrupted because I do a lot of intercontinental traveling, it doesn't bother me. It doesn't affect me. Partly it's because I don't get jet lag. That's just luck, really. And also I can sleep really well on planes. So some people can't. I mean, most people can't. So yeah, different things work for different people, and that's really what Bryan is emphasizing when he says measure, measure, measure.
So just to kind of sum up this part of the discussion, it does seem that certain habits does delay biological aging, but we are not sure by how much because we're not certain about the accuracy of the measurements, correct?
We're not sure about how much because we're not sure of the accuracy, and also we're not sure how to define it. So as I said, it's rather questionable whether the impact of losing weight should be counted as within this framework. But the reason why people tend often to do so is because there are these indirect knock-on effects on the whole of our metabolism, on things that really feel like they ought to be included. And it's the same with stress. If you have a stressful life and you're not good at handling stress, that's definitely life-shortening. It has an impact on stress hormones like cortisol, of course, and these have effects throughout the body. So if you can adjust your life or your response to stress, just be more chill, then it's going to be good for you. But again, is that really changing your biological age? By some definitions, yes, because it's changing things in your physiology. It's changing the rate of accumulation of damage. But, other people would say, well, this is just lifestyle. It's not really changing your age.
When you look at kind of the race to longevity escape velocity, for example, when I read Kurzweil, it just seems like it's something inevitable that's happening. Like Earth is this machine making intelligence and technology. Another way of looking at it is saying we need that brilliant one person, perhaps yourself, who makes this major breakthrough, and then it happens. And if we don't have those individuals, then it will never happen. But how are you thinking about it? Will it happen eventually, or is it up to you or someone else?
I guess I've got two main answers to that. Historically, as in like before this century, we could say in a very abstract sense that yes, it's inevitable. The body is a machine. We knew that already. The body is a machine. Its function is determined by its structure. Its longevity is determined by the extent to which it damages itself or it accumulates damage. And therefore, in principle, we definitely can keep the body going and transcend its warranty period, if you like, simply by sufficiently comprehensive preventative maintenance. But at that time, people were not thinking about it that way. So, of course, the big contribution that I made back in 2000 was to come up with saying things in that way, saying, listen, we just need to develop preventative maintenance, and making it concrete by enumerating these 7 categories of damage that each had a corresponding generic approach to eliminating that damage. So from then on, it became possible to think in terms of progressive improvements in how comprehensive the elimination of damage can be. And that immediately led, only a few years later, to the concept of longevity escape velocity. Ray was certainly thinking, pretty much independently in the same along the same lines, and he started using the phrase "live long enough to live forever" in the same year that I started talking about longevity escape velocity. So, yeah, so it is absolutely inevitable, no question. It's just what's not inevitable is the time frame.
Yeah, so the right person could make it happen 5 or 10 years before.
So, yes, but the right person is not a scientist. The right person is a billionaire. Because the fact is, there are a lot of different technologies that need to be developed. And we need to do that, and that's a lot of people. Yes, we had to come up with the concept of damage repair as the way to go, but that's done now. That's done. It took me maybe 10 years to get that really into the mainstream within the expert community, but it's certainly there now, and it's it's pretty much mainstream within the wider world as well. But yeah, I mean, that still means a lot of work by a lot of different scientists with different areas of expertise. And of course, it's all got to be put together as well. So, I mean, my take is that it just needs the funding. Now, when I say it needs a billionaire, it doesn't really even need that. If you've got 100 million, you could do it. Because actually, what we really need to do is mobilize the public. Once we once the public is starting to demand that we, try to defeat aging medically as soon as possible, governments will do it because governments like to get re-elected, right? And so, that's why I've been working on the public for quite a long time. Of course, we haven't made much progress yet. The public is still overwhelmingly in this, what I call the pro-aging trance, where it tries to make excuses for aging being some kind of blessing in disguise, for the sole purpose of putting aging out of people's minds, of course, and letting people wanting to get on with their miserably short lives and make the best of it, rather than face up to this ghastly thing that is going to happen to them. But we're getting pretty close to being able to make that sea change happen, I believe. I think that the experiments that we're doing at LEV Foundation, once they reach fruition, in other words, once we get this doubling or trebling of the effect size that can be achieved at the moment, every expert is going to completely know that we are in a very new world. It will be a complete, it will be a qualitative change. It won't just be incremental progress. And they'll say so. They will say so publicly. That's what we're going to need in order to get the influencers of the world, Oprah and MrBeast and, Rogan and so on, to actually, I mean, these are people who are already interested in aging. They say things about it. They have people like me on their shows. But they're not interested enough to come out and demand a war on aging, right? And the reason they aren't is because they haven't seen enough or heard, or more precisely heard enough from the subject matter expert, to believe that now's the time. At the moment, you see a lot of, warm words from the scientists saying, oh, yeah, we think we know how to do this. We think we discovered this and so on. But until it turns into actual concrete progress at the level of mouse lifespan, they're not going to be very interested. And so I believe we're pretty close. I think, within the next few years, we've got a very good chance, just as long as we get the funding, of actually getting to the point where the expert community in general, not just me, will be out there saying, we've made a qualitative breakthrough here, and then everything's going to happen after that. So in other words, even though it's going to cost trillions of dollars to bring aging under control at the as quickly as we actually could, it's only going to cost like 20 million to trigger the release of those trillions of dollars. And we know this now because of COVID, of course. You may have thought, 10 years ago, well, the public are just apathetic. They just don't do things. But when there's something that the public actually acknowledge is an emergency, is a rather bad thing, then, no government got, thrown out of power for putting a lot of taxpayers' money into defeating COVID, right? So it's going to be a COVID scale, or in fact, it's going to be a COVID eclipsing war when we get this to happen. But it's only going to take $20 million to trigger it.
Well, we're actually seeing a little bit of a difference here in Denmark. The book I wrote together with the author Lars Tvede was released. It's now 3 days ago. So we got, first of all, we got in some of the biggest podcasts in Denmark talking about longevity escape velocity. The hosts there were actually quite open, saying, I actually agree. I also think it's going to happen, which is a new thing. We got on the biggest TV station, national, the morning show there, also talking about the book and longevity escape velocity. And I just saw a review in the largest newspaper in Denmark, giving the book 5 stars and actually mentioning that, we're going to live 250 years and beyond in the headline. So it's interesting. I mean, on a small scale, Denmark is a small country. I think it has gone from people saying you are more or less crazy when I talk about it, till, really starting to engage in discussions, and some people even being more aggressive in when they think this will happen than myself. So I can actually see when you put in the work, you can change people's mind.
Yeah, totally. And I'm saying this worldwide. However, it needs to go further. You've got certainly, you're absolutely right to be saying that this is progress. This is big progress. That, and the way I would put it is, generally, the media treatment of me personally has become very much more, I would say, respectful, serious. Historically, with plenty of media treatment of me 20 years ago, I was everywhere, but it was really entertainment. It wasn't, it wasn't done in an educational way. And yeah, so the whole thing has become much more in the much more serious, and that's definitely progress. But it doesn't rise at this point to the level of, for example, the German longevity party actually, getting seats in the Bundestag, things like that. Now, Denmark, of course, may play a punch above its weight, shall we say, play a disproportionate role in the near future on account of its association with Ozempic, which is, the first thing since statins, really, the first thing for 50 years or so, that can be genuinely called a preventative medicine for age-related conditions. And of course, there's plenty of work going on right now showing that, very much as I was saying earlier in relation to damage repair, that the benefits of weight loss achieved by Ozempic are quite broad. There's lots of knock-on effects on things that are not necessarily linked in most people's minds to weight loss. So yeah, There's a certain amount of national pride here as well that probably helps, in very much the same way that Japan started to actually pay attention to regenerative medicine after Yamanaka discovered the factors that could create induced pluripotent stem cells nearly 20 years ago.
So, outside of what you're doing yourself, what would you say kind of are the most significant contributions to longevity escape velocity? Is it the Yamanaki factors, or is it what Bryan Johnson is doing? I guess is what Bryan Johnson is doing is it kind of a mouse study on a person?
Kinda. Actually, yes. Brian Kennedy, one of my friends who's also a very prominent gerontologist, has got into the habit of saying that we're lucky in aging research right now that we have a new model organism called billionaires. Um, uh, um, um, so, you know, I mean, as I say, Brian's definitely making an important contribution. And there are plenty of contributions coming from laboratories around the world around, developing new technologies that can be used for this. Most of those technologies tend to be ones which actually have a rather broad applicability across medical progress in general, not just against aging. Certainly, induced pluripotent stem cells is an example of that. So is CRISPR. I would say if I had to pick one thing outside of our own work that is going to make a really big contribution, it has to be artificial intelligence, because artificial intelligence is, already making a huge difference in the speed at which we can develop drugs and interventions. We started out with AlphaFold not long ago, but even before that, there was plenty of progress using machine learning to identify promising drug candidates. A company named In Silico Medicine, run by Alexei Boronkov, great friend of mine, has done very well in that regard. It's not the only one. But it's going further. So we've now got Alpha Proteo, which is a kind of successor to AlphaFold. It is a program that doesn't simply predict the three-dimensional shape of a protein from its amino acid sequence. It does the reverse thing. You start out with a shape that you want, and it figures out an amino acid sequence that can provide it. And we've got examples of this kind of thing working already. Just last week or the week before, there was a big announcement from Retro Biosciences in their collaboration with OpenAI, where they figured out ways to change two of the Yamanaka factors, not just a little bit, but change, I think, something like 50 amino acids in the sequence. And the result was versions of the two proteins that could work just as well at 50-fold lower concentration, if I remember the numbers correctly. So, this is the kind of thing that evolution can't do, because evolution always goes in small steps. And yet, in silico, we can do it. So this is fantastic. Now, I do want to, before I move on away from AI, I want to caution, though, because at the moment, there are some people, including some people who are billionaires, who are making a huge, huge mistake when it comes to AI. They're saying, okay, AI is improving really, really fast. It's going to be pretty completely omnipotent very soon. Therefore, let's not bother with doing any experiments anymore, because we can just sit back and let AI do it for us. And of course, we can spend that money and time hastening the arrival of that AI, and we'll probably make plenty of money out of that as well. So that's all great. But the problem is, AI does not generate data out of thin air. It generates insights from data. And therefore, the best that AI could possibly do, even once it was arbitrarily omnipotent and clever, is it could tell us what experiments to do, or it could do them itself. But the point is, it's still got the experiments have to be done. And how fast those things are done depends on people. So, we've really got to do those experiments. And the sooner we actually get on and start doing them, whether or not we've already got the AI, the more lives will be saved. So I believe that at the moment, there is there is a constituency that is excessively enamored of the potential for AI to solve everything. And we need to be have a bit more of a sense of proportion about that.
So, do you think will there be a day where maybe you could do an actual mouse study, but then you could amplify the mouse study in AI, so kind of simulating 1,000 mice or 1,000,000 mice, while in your physical study you only have 10? Is that a possibility?
I don't think so. In fact, more generally, I think that the whole idea of doing biological simulations of anything bigger than a cell in silico is not going to happen for a very long time. And the reason I say a long time is not because I think it's going to take a long time to get the computing power or the AI cleverness or anything. I don't think that's going to take very long at all. The problem is collecting the data. Again, AI did not generate data out of thin air. If you're trying to simulate a physical system whose components you understand right down to the atomic level, then sure, it takes compute power and smart. It takes AI, and that's fine. There's a lot of things that we will be able to simulate really precisely and really well. Problem with the body, well, any living system bigger than a cell, is that we don't have the data. We don't even know enough about the composition, the structure and composition of a living organism. So we can't give the AI the starting point it needs, and there's no way to get that information without doing experiments in the real world.
So let's try and talk a little bit about the mouse studies, and also maybe explain to people about longevity escape velocity. When you're talking about like comprehensive repair and damage, is it a fair way to explain the body that you have like a few time bombs that can go off, like your brain, maybe your heart, your liver, or what's the best way to kind of understand why we die and what it is we need to repair?
Well, I often use the analogy of simple man-made machines like cars. I simply point out that the human body is a machine, same as a car is. The human body is vastly more complicated, but that's just a difference of degree. So why do cars not last forever? Well, basically because in the process of being a car, you accumulate damage. You accumulate rust and contamination in the oil and other things. And some of those things are fine because you can fix them by the annual maintenance that you get, that you do with your car, that's demanded by the law. But some of them are not repaired as a result in the course of that kind of maintenance. And so the car keels over after 10 or 15 years, except when it doesn't. As we know, we have some cars that are 100 years old. And they didn't get that way by being built unusually well to be designed to last 100 years. They were designed to last the same length of time as the ones that only lasted 10 or 15 years. But they got an exceptionally comprehensive amount of preventative maintenance, more than what the law requires. And the result is they're working every bit as well as when they were first built. Even though they're 100 years old. And everybody knows that if you carry on doing that, there's no limit, right? If you asked someone 100 years ago when they were building such a car whether there's any chance that the car would last 100 years, they'd have laughed at you. But if you ask someone now who owns such a car whether it's going to last another 100 years, they'll say, of course, why not? We'll just carry on doing the same things, the same preventative maintenance. So it's the same for the human body. The human body is not subject to all that much medical preventative maintenance, but it does a lot of its own preventative maintenance, as we were talking about earlier. So we only really have to fill in the gaps. There are certain types of damage that accumulate so slowly that the body, the evolution hasn't taken the trouble to invent automatic built-in preventative maintenance for those things. And those are the things that cause the eventual decline in physical and mental capacity and eventual death. So we just have to augment our built-in damage repair machinery with machinery that we can develop medically. And the better we get at that, the longer we can keep going. Now, the good news, and this is where the concept of longevity escape velocity comes in, is that we don't need to develop all of this in one go. We already live 80 years or thereabouts. Therefore, if we can develop preventative maintenance that is not completely 100% comprehensive, but is comprehensive enough to give us maybe 20 years of extra life, then that buys us, when I say us now, I mean the research community, those 20 years to do better, to develop damage repair 2.0, which will be better at repairing the types of damage that were still not repairable by damage repair 1.0. And so we can kick the can further down the road, and we can carry on doing this as long as we like. So basically, my belief is that once we get those first 20 years of extra life, we will be able to, we've done the hard part. We will essentially have no reason to ever have people suffering from the health problems of late life, however long ago they were born. And, that's because damage repair is not done to kids. You do it to people who are already in middle age, maybe 60, and who therefore have accumulated as some damage that needs to be repaired.
But what would the typical things be that kill us? Like, just if we keep the car analogy, I guess the gearbox would be one thing, and as you mentioned, in some countries it would be rust. And in some countries, I guess it would more be the engine. So it all depends, right? If we look at humans, is it mainly the organs and the brain that would be the ones that basically kill us?
Well, so, I mean, as with any machine, the components of the human body all talk to each other. But because the human body is so very, very complicated and has, been designed by this very, hit and miss process called evolution, we've ended up with a situation where the amount of crosstalk between different parts of the body is even more sophisticated than it is in a simple man-made machine. So I don't really like to think of it in terms of, let's fix a few organs and we'll get 20 years, because that's not how it's going to work. You have to fix the whole body fairly well. You don't have to fix anything completely, but you have to fix the whole body fairly well. And the good news is that all parts of the body are made of more or less the same underlying components. Everything's made of cells and stuff between cells. And, cells are made of the same stuff, DNA and proteins and so on. So the kinds of treatments that will work in one place in the body will also work with not very many modifications in another place. That was what really gave rise to the 7-point classification that I came up with 25 years ago. That, let's take loss of cells. When cells die and they're not automatically replaced by cell division, the number of cells goes down, some cells of that type goes down. And that is a type of damage, because eventually you have too few cells for the job that the cells are supposed to do. So where does this happen? Well, one place it happens is in the brain, specifically in one tiny part of the brain called the substantia nigra, which has a type of neuron called a dopaminergic neuron. Those neurons, because of the work they do, making dopamine, they have a very stressful life, so to speak, and sure enough, they die at a much more rapid rate than other neurons in the brain. So by old age, all of us have lost maybe a quarter of the dopaminergic neurons that we had when we were teenagers. But that's actually okay, because the body is set up to tolerate losing that amount. Problem is, of course, that different people lose these neurons at different rates. Some people end up losing maybe 3/4 of them by old age, and those are the people who get Parkinson's disease. So the right way to fix that is stem cell therapy. You just put cells in medically that will divide and differentiate to replace the cells that the body is not replacing on its own. And there are half a dozen clinical trials going on now to do exactly that. It was first done 30 years ago, actually in Scandinavia, in Sweden, And it was only very occasionally successful because back then we didn't have induced pluripotent stem cells. We didn't have we didn't have 1% of the knowledge we have today of how to manipulate stem cells in the laboratory before injecting them. So it was extremely hit and miss. You had to use cells taken from something like the right part of the brain of an aborted fetus. Now, people are very hopeful. But that's just the brain. So you may say, well, hang on, you've got to do stem cell therapy elsewhere as well, like in the heart. So, for example, there is a type of cell called a pacemaker cell, which is responsible for ensuring that the heart keeps beating. We lose a lot of those. Again, you need stem cell therapy. And you need a different stem cell therapy. You don't want to give them dopaminergic neuron precursors, right? But fine, because even though different stem cells are different, the technology, the development of a particular type of stem cell, of different types of stem cell, has an awful lot in common. You start out with a pluripotent cell, for example, and so you can reuse the same knowledge. Same thing with the thymus, for example, which is an organ that is responsible for part of the immune system and which atrophies during life. So all of these things can kill us in different ways at the level of the actual pathology. But the technology to fix them is very much in common across that one category, and the same applies across each of the other 6 categories.
But will that mean that, we might get technology first to fix the brain, so we'll not die from, for example, Alzheimer's? But, the final thing we have a challenge fixing would be the liver or something like that.
Yeah, so I don't think that's going to happen. I don't think that's going to be, it will be like that in a way, but the time frames will be very, very slight. In other words, by the time we are able to fix, let's say, the heart really well, we will be like within months of being able to fix the brain as well. Or in whichever order it's going to be. Simply because by that time, once we have a proper war on aging, because we've had success in mice and so on, it will be all hands to the pump. Everyone will be doing their own thing, working on things and talking to each other and getting information from each other. And because of this enormous overlap and intersection between, for example, different types of stem cell therapy, once you can do one of these things, you're going to be very, very close to being able to do the next one and the one after that.
Can we just go through the 7 areas that we need to repair?
Sure, yeah. So the one I've just talked about is cell loss, where cells die and they're not automatically replaced by the division of other cells. And so stem cell therapy is the obvious way to fix that. We now, of course, also have this thing called partial reprogramming, which is essentially using the Yamanaka factors in vivo to make cells that are fairly stem-like already a bit more stem-like. And that's something that probably can stand in for stem cell therapy in a lot of places in the body, but not everywhere. I don't think you're going to be replacing your dopaminergic neurons using partial reprogramming. So we may end up being able to make the problem simply using partial reprogramming, but we won't solve it entirely. What's the next one? Next one, well, let's take cancer. I'd actually like to use a slightly more general term. I call it division-obsessed cells. So cells that divide when they're not supposed to. So thing about cancer, people often say, cancer's not just one disease. Different cancers are so different from each other. We need to tackle them in a very customized way. And certainly customized cancer treatments show great promise. We have this thing called CAR-T, which was developed not long ago now, only about a decade ago, and it's transformed a lot of things. The idea of using the immune system against cancer or making the immune system work better against cancer than it normally does has been around for decades and decades and made basically zero progress for God knows how long. And then suddenly there was this huge breakthrough, and almost simultaneously another breakthrough called checkpoint inhibitors, which essentially allowed the immune system to work better. And there's still a long way to go there, but it's going well. However, What we'd really like is an off-the-shelf, truly universal cancer therapy. And so we have to say, well, how can you have a universal cancer therapy when all cancers are different? Answer is that saying all cancers are different is an oversimplification. Cancers have distinctive features, but there's one thing that all human cancers have in common, and that is they all have a system for extending the telomeres, the ends of the chromosomes. If they don't, and the reason they have that is because a cell that doesn't have a system for extending the telomeres has its telomeres shortened every time it divides. And of course, the definition of cancer is cell division, uncontrolled cell division. So the cancer just will never get big enough, anywhere near big enough to kill you, without having to turn on some kind of telomere extension mechanism. And 90% of cancers in humans do it with this one enzyme called telomerase. So I've been interested in somehow disrupting the ability of a cancer to do that. And right now, we have a drug that does exactly that. It's an extraordinary discovery made by a great friend of mine, Jerry Shea, in Dallas, Texas, some time ago, now in clinical trials in Australia, overseen by a company named Maya Biotechnology. It's a drug that basically, well, to use the technical term, it turns telomerase into a suicide gene. So to explain that in non-technical terms, basically what happens is that this drug kills cells that are making a lot of telomerase. And, it kills them quickly. So it's, and I could go through the mechanism of how it does it, but the point is it does it really well. And especially in combination with a cancer immunotherapy, it's mind-blowing. So a big tragedy right now is that there's just this one clinical trial going on, relatively small number of patients for one type of cancer. We should be doing 100 clinical trials in parallel on different types of cancer, just using this one drug. And, so anyone who does want to throw, $1 billion at this problem rather than only the 20 million that I need, that's where I would put it. So that's number two. And the reason I say division-obsessed cells is because there are things that involve the clonal expansion of cells in a subtler way, and that may be important as well. So there's something called myeloid skew. Let me explain briefly what that is. So in the bone marrow, of course, we have these stem cells for the blood that constantly generate the blood. And they are, there's a lot of different types of cells in the blood. I mean, we say, obviously, there's red cells and white cells, and then there's platelets. But there's a lot of different types of white blood cell. And biologists group these cells, these many different types of cell that the blood has, into two major categories called lymphoid and myeloid. And it turns out, for whatever reason, that progressively through life, myeloid cells become more abundant, lymphoid cells become less abundant, and this is bad for you. You need lymphoid cells. People in Stanford, in Palo Alto, last year or year before, discovered a really cool way to reverse that process, essentially by killing off some of the myeloid cells and giving the lymphoid cells more opportunity to proliferate. And it seems to work really well, in mice anyway. So, this is another example. The third one is another one that many people in the audience will be familiar with, which is senescent cells. So here we are talking about cells which, again, are bad for you, but they are not bad for you because they divide excessively. They're just bad for you because they hang around and they're toxic. They secrete nasty things. Now, these cells, people figured out, very much to my surprise, maybe 15 years ago, that there are drugs that can selectively kill these cells. And the first generation of those drugs were not selective enough. They were not really the real deal, but we're getting extremely good now at selectively killing off these senescent cells. They are things that, again, could potentially be eliminated by the immune system. The immune system does eliminate most of them when they arise, but not all of them. So again, people are also looking at ways to make the immune system better at doing that. But honestly, it looks as though it probably won't be needed because because drugs can do it instead. So then if we go inside the cell, we're talking, so far I've just talked about whole cells, right? So if we go inside the cell, then there are two major types of damage. So the first one inside the cell is mutations in the organelle called the mitochondria. So all cells, almost all cells, have a lot of these things called mitochondria, which are responsible for extracting energy from nutrients and using it to do all the things that the cell does that need energy, like, building proteins and replicating DNA and so on. Mitochondria have their own DNA, and that DNA mutates much, much more rapidly than the DNA in regular chromosomes. We have to do something about that. And there are various approaches that might work. One way is to make copies of that DNA and stick those copies in the nucleus. In our regular chromosomes. And I've been trying to do that for about 20 years. It's our longest standing research project. And we've made a lot of progress, but it's very, very difficult. There are a lot of reasons why it's hard. And so we may never get it to work, to be honest. Therefore, we'd like alternative approaches. And, well, essentially, most of the alternative approaches revolve around tweaking the way that the body recycles mitochondria. Even in cells that are not dividing, mitochondria are constantly recycled. Now, some of them are destroyed and other ones are replicated. And essentially, the balance between those two processes, the replication and the destruction, is what causes the problem. Mutants arise that somehow have a selective advantage and they expand, a bit like a cancer within a cell, if you like. And they take over the cell. So there are various ideas now for how to reverse that selection, so that the mutant mitochondria have a selective disadvantage and they never accumulate. Or even if they've already accumulated, that the expansion goes into reverse. But those are ideas that are at a pretty early stage of development right now. Okay, so the next one inside the cell is a nice simple thing to describe. It's just garbage, waste products. So the body makes a huge number of waste products all the time as byproducts of the things it needs to do. And nearly all of those things are destroyed as soon as they are created. The cell has a nicely diverse system for doing that. But there are a few things that the body doesn't know how to destroy, that simply accumulate really slowly, so evolution hasn't taken the trouble to invent ways to get rid of them, either by excreting them out of the body or by just breaking them down. And these things accumulate typically in a part of the cell called the lysosome, which is a kind of incinerator that can incinerate most things, but not everything. So basically, we need to improve the incinerator. So we've been trying to do that. It turns out that you can use the living world quite well in this regard. You can identify enzymes in bacteria or in fungi in the soil that already destroy things that we can't destroy. And then you can put the genes for those enzymes into cells and make them destroy things that they would not normally be able to destroy. This has ended up working really quite well for a material called A2E, which turns out to be the thing that causes macular degeneration, the number one cause of blindness in the elderly. This is a chemical that's created as a very occasional byproduct of the chemistry of vision, the chemistry of seeing stuff. And it accumulates in cells called the retinal pigmented epithelial cells at the back of the retina. So we've found enzymes that fix that. I passed this project on to a spin-out company out of one of my nonprofits about 10 years ago, and they've continued to progress with it, and it's now working well enough that it's in the process of being sold to Big Pharma, so that's pretty good news. The other alternative, as I just alluded to, is rather than destroying the material in question, you can get the body to excrete it. And that's pretty difficult in the eye, but it's not so difficult in the bloodstream, and in particular in the circulatory system. So in the major arteries, we have material that's, again, not destroyed when we'd like it to be. And it is basically oxidized cholesterol. And so another group that used to work for me and now have a spin-out company is pursuing doing this, basically extracting oxidized cholesterol from the artery wall and causing it to be excreted out of the liver. And so, so this is a big part of the gaps, if you like, in inbuilt damage repair machinery, the things that we need to fix. So then finally, there are two types of damage that occur outside the cell. One of them is, again, waste products. So the most famous one of these is senile plaques in Alzheimer's disease. But the amyloid, as it's called, that accumulates there, accumulates in other parts of the body as well. It was discovered not all that long ago, 15 years ago maybe, that in the heart, you get an accumulation of a different type of amyloid composed of a different protein called transthyretin, which seems to be really bad for you when you get to be over 100. So we've got now ways to break that down. There's a special type of antibody called a catobody, which doesn't just bind to some antigen; it actually chops it up. It cleaves peptide bonds. So this is now very much in progress. Another spin-out company is working on that. And then finally, there is cross-linking. So again, coming back to the arteries, the cholesterol I was mentioning is the cause of atherosclerosis, which causes heart attacks and strokes and so on. But we also have this thing called arteriosclerosis, the hardening of the arteries, which leads to high blood pressure and lots of knock-on effects of that. And that's basically a loss of elasticity of the arteries. And it's the same chemistry that goes on there that causes wrinkles in the skin, loss of elasticity there, and also, for that matter, presbyopia, so long-sightedness, inability to focus on things close up. It's all to do with chemical reactions between proteins and sugar in the circulation. And these chemical reactions cause new chemical bonds to form between proteins that are supposed not to be bonded together and supposed to be able to slide against each other. So this is where the stiffening comes from, the loss of elasticity. And so now the goal is to develop ways to break those bonds. And it's going quite well. Again, there's a spin-out company working on it that comes from the work that we funded a long time ago. So yeah, all of these things are moving forward, but it's still, in many cases, a long way to go.
That was today's Mind Blowers with Aubrey de Grey. We'll continue to follow rejuvenation technology closely here on the Mind Blowers podcast. See you next week. And if you'd like more insights into the future, check out one of our other podcast episodes right here.